or MIF4 for short, in which it seemed that proteins were being imported to the matrix space but then failing to fold into their native state. And they said, well, it sounds interesting, but you have to consider the idea that the proteins could be jamming in this contact site between the two mitochondrial membranes during import, and if the N-terminus of the protein goes through, the leader peptide gets clipped and the protein's just stuck in the membranes. And we said, well, it surely doesn't look like that because the proteins are not being cleaved in any other way that we can see other than through the signal peptide. But we'll send you the strain, and you should look with your good in vitro biochemistry with isolated organelles to see what's happened to these proteins. 2 weeks later, Ulrich called back very excited and said, you're absolutely right. We've made isolated mitochondria from these from the MIF4 strain carried at non-permissive temperature with a variety of import reactions carried out both in vivo and also in vitro. And the proteins are completely inside the matrix. We can add exogenous protease to the mitochondrial organelles, and it doesn't touch the proteins. So we very rapidly, together with them, looked at a host of other proteins, all of which proved to be affected in terms of their biogenesis. They all failed to reach the native state. But most exciting to all of us was the risky iron-sulfur protein, because this protein is a monomer in the mitochondrial matrix during its lifetime there. It comes in as a monomer, and then it's cleaved twice through its signal peptide before it finally lodges in the inner mitochondrial membrane. And what we all observed was that there were no cleavages taking place at all. The iron-sulfur protein made it to the matrix, but failed to be cleaved and seemed to be in an insoluble aggregate. So this suggested that the activity that was getting affected was not necessarily oligomeric assembly, but maybe the step before it, namely protein folding. And so Ulrich then, in collaboration with Joko Mosterman, carried out another more pointed experiment with dihydrofolate reductase, where he sent DHFR into mitochondria by attaching a signal peptide to it, and he could show that the protein bound to a large complex when ATP was deprived from the system and occupied a non-native state that was sensitive to antibodies specific for the non-native state and was also very protease susceptible. If he then added ATP back to the system, he saw the DHFR get released from this complex, and it now assumed protease resistance like the native state and appeared to have reached the native state. So this suggested that whatever this mutant was that we were studying, it had something to do with polypeptide chain folding in the mitochondrial matrix space. And so Ming very rapidly, with a yeast genomic library, rescued this mutant, and the gene that came up corresponded to a heat shock protein that had been identified the year before by Richard Hallberg working in Iowa in T thermophila mitochondria, a roughly 2- or 3-fold induced but quite abundant protein in the mitochondrial matrix space of those organisms. So we called Hallberg up on the phone because there was a rumor that he was sequencing the yeast version of that protein, and indeed we could sit there and match the DNA sequence 1-to-1 of the gene that rescued Ming's mutant and the HSP60, what we all dubbed collectively as being heat shock protein 60, that he was busy sequencing from Baker's yeast at the same time. So HSP60 is a bit of a misnomer for this molecule. It's essential at all temperatures, suggesting that its ability to assist protein folding in the matrix space is critical under all conditions. I should point out that studies from Sabine Rossbert and others have suggested that roughly 30 to 40% of imported mitochondrial proteins absolutely require this machine in order to reach the native state. Furthermore, HSP60 requires itself in order for its newly imported subunits to be assembled into new complexes inside the mitochondrial matrix, which is consistent with old data that mitochondria beget more mitochondria. They're not made de novo. So this is an early picture of Ulrich and his wife Manojit and me during a trip to the Black Forest, where we were just walking around speculating on what could this large assembly do? How could it do this? Was it a back-to-back set of rings? Where did polypeptide bind? How could this work? What would be a role of ATP? They were all just wide open questions. I can't recall what book it is that Monagit's actually reading to us, but maybe she's already predicting how things were going to work. In any case, HSP60 in mitochondria was not the only double ring machine around that would be termed a chaperonin. It was part of a family called the group 1 family that included GRO-EL in the bacterial cytoplasm, identified literally a decade and a half earlier by Costa Georgopoulos as a cellular gene whose mutation allowed lambda phage infection to proceed without lysing the cell. In other words, lambda was only abortively able to infect bacteria that were GRO-EL deficient. And in fact, Georgopoulos opened up the mutant cells, and what he saw were these monstrous-looking, misshapen phage particles inside of his infected GRO-EL deficient bacteria, suggesting that GRO-EL had some sort of role in oligomeric assembly. He also noted that this gene appeared to be critical to cellular growth, suggesting that it had something to do with more than just phage infection, but with behavior of host proteins, perhaps, or host metabolism. In addition, in the late 80s, or 1980, to be more exact, John Ellis and co-workers working in Warwick identified another double ring assembly inside the chloroplast stroma that appeared to interact with newly made large subunits of Rubisco, but was not present in the final L8S8 hexadecamer that's mature Rubisco inside the plant chloroplast. And he thus implicated the Rubisco binding protein in assembly of Rubisco, the most abundant protein in the biosphere. But I think our work in mitochondria shifted the paradigm of how these machines might be working to one of polypeptide chain folding. And I'll just briefly mention that we looked very shortly after these initial experiments for versions of chaperonins in other kingdoms. And in fact, Jonathan Trent, who came to the lab and worked with us and with Horwich as well, identified a double ring assembly composed of 9 subunits per ring and sulfaloba shabatii that conferred thermotolerance when it was taken to a high temperature. When we sequenced this particular gene, we saw homology in what proves to be the ATP binding site of this component shared with the ATP binding sites of the type 1 chaperonins. But even more strikingly, this component bore a striking resemblance through its primary structure to a component in the eukaryotic cytosol that had been in search of a function called TCP1 complex, or otherwise known as CCT or TRIC or what have you at this point, which is a heterooligomeric double ring assembly that's essential for folding actin, tubulin, and a host of other proteins, including particularly beta propeller proteins. And so these type 2 chaperonins look fairly similar to the type 1s in being back-to-back double rings, but they contain their own built-in lid structure that's involved with encapsulating proteins that they're going to mediate the folding of. And a recent paper from Judith Friedman's group has suggested that in fact their ATP cycle is slightly different, that hydrolysis is actually used to close their chambers, which differs a little bit from the type 1s. So I'm going to focus the rest of my talk on GRO-EL as being the sort of canonical chaperonin from which we've gotten the largest number of lessons on how these machines might be working. So the first reconstitution of a chaperonin reaction was carried out by George Lorimer and his co-workers while at DuPont. And what they did was they took a Rubisco from Rhodospirillum rubrum. That's a dimeric form of Rubisco composed of 2 identical 50 kD subunits. And they showed that if you diluted the R. rubrum Rubisco from denaturant, the protein just crashed out of solution and never reached the native state. By contrast, if GRO-EL was present, a binary complex was formed that they could show was a stoichiometric complex. 1 molecule of Rubisco associated with 1 tetradecamer of GRO-EL, GRO-EL being 2 stacked 7-member rings composed of 60 kD subunits. And in fact, this localization in the whole is really work that Ulrich and Wolfgang Baumeister did, as well as work that our lab did with Jim Hainfield and Joe Wall at Brookhaven. They showed by negative stain EM that there were masses in the hole. We gold labeled a DHFR and could show in the stem that the gold particles are localized in the central hole of the machine. In any case, in the second step of the Lorimer experiment, they added the co-chaperonin GROW-ES, a 7-member ring composed of 10 kD subunits, and over a period of minutes, they observed the production of native rubisco. So the field was really focused at that point both on how polypeptide binding prevents aggregation and on how this step of GROW-ES association and ternary complex formation could lead to productive folding. And so before going further, I want to just assure you that Anfinson's principles were alive and well through all of these initial experiments. I think from the very beginning, all of us believed that the idea was that chaperonins provide kinetic assistance to protein folding. That is, as Peter mentioned, under cellular conditions, there's very high protein concentration. A temperature of 37 degrees represents a relatively high temperature for many proteins in terms of their productive spontaneous folding. And so the idea, in a nutshell, if you don't listen to another word I have to say here, is that binding to an open ring of a chaperonin essentially pulls a non-native protein up the energy landscape out of what would be in the rugged landscape of the cell, out of a kinetic trap, and gives it another chance to try to get down the landscape to the native state. We believe, and there's not complete agreement in the field, I would say at this point, but we believe that folding in an encapsulated chamber basically lies on the same energy landscape as free solution.